Hi, this is podcast number 2. And what we're going to talk about in this one and the next one is some COVID-19 background. So, obviously, we're in the middle of a pandemic. You are taking a cell and molecular class, and there is nothing more cell and molecular than this pandemic. I think that knowledge is power, and I want to give you some tools for understanding what's going on in the pandemic. And as we approach different subjects walking through this class, we're going to run into things that pertain to this pandemic. I talked a lot about this in another class I teach, cell physiology. Some of you took that class before this class. It's sort of designed that this class should come first, but it doesn't matter. This class is more like an anatomy class, and that class is more like a physiology class in that how the different parts talk together. But again, I think that most of the classes that you take from biology this semester are going to be talking about the coronavirus. And it's really important that you understand what's going on as a biologist and talking to people who are not biologists about what's going on, because there's also a lot of misinformation being spread. So with that in mind, I'm going to give you a brief overview of some background on the coronavirus pandemic. And then as we approach different topics and there's something related to coronavirus biology, when we're covering other things during the class, I'm going to bring it up. And you should ask questions. I've been on the radio 6 times since the pandemic has started talking about this. I feel like if there's any way that I can contribute to making this better or making people understand it better, then I should, and so should you. So please ask questions so we can spread good knowledge about what's going on with the coronavirus. Okay, so when I say coronavirus, that's actually a family of single-stranded RNA viruses that look like this. So you've seen some version of a picture of this. Corona means crown, and that's to do with these spike proteins. And there's a whole family of these proteins. And it's very likely, in fact, almost assuredly, that at this stage in your life, you have been infected by one of these viruses in the past. And normally they cause a common cold, much less severe symptoms than what we're seeing now. COVID-19 got its name from the disease caused by the novel coronavirus. So CO is corona, vid for virus, V for virus, and D for disease. So coronavirus disease, first discovered in 2019. And then it's often referred to as SARS-CoV-2 when you get into the primary literature. This is something that you see a lot. So SARS-CoV-1 was the epidemic from 10 or so years ago that caused death in mostly China, a little bit in Toronto, and that was SARS-CoV-1, a very similar genetically virus to this virus, and this is referred to as SARS-CoV-2. You'll see a lot of papers talking about what we know from SARS-CoV-1 and the survivors of SARS-CoV-2, and can we extrapolate any of that information into understanding what's going to happen with SARS-CoV-2, because we're still in the middle of this. Okay, what happens is that there is a receptor in your cell membrane. These are ACE receptors. That ACE stands for angiotensin converting enzyme 2. There's another co-receptor called TMPR1 that SS2 that can also contribute to binding the SARS-CoV protein. And it's the spike proteins on the outside of the virus that are recognized by ACE2. So these ACE2 receptor proteins are found in the cell membrane of cells that line your respiratory tract, so the inside of your mouth and your throat and your lungs, in your nasal cavity. It's thought that this just came out this week that the reason why some people lose their sense of smell is that there's a particular part of your nasal passages that is really, really high expression of these receptors. And that is connected to your olfactory bulb in your brain. And it's thought that because there's so many receptors in that particular part of the nasal epithelium, that is like a big bullseye for this virus and how the virus enters the body. So this is an enveloped virus, meaning that it's surrounded by a membrane. That envelope virus gets incorporated using your own cell endocytosis machinery. And we're going to go over this in detail when we talk about membrane traffic. When it gets incorporated, it releases its genome, and the genome is a plus strand of RNA. So what that means is that it's the same orientation and the same configuration as messenger RNA. So the messenger RNA, which you've learned in other classes, gets read by ribosomes and gets translated into the protein. This virus has a messenger RNA that is ready to go and is ready to be read by your ribosomes. And then your ribosomes convert that viral mRNA into two big polyproteins. So we normally think of one string of mRNA making one protein. In some bacteria and viruses, what happens is it makes a giant protein, then gets cut into separate proteins. So one long string of amino acids that gets cut several times by an enzyme called a protease, and then they fold into separate proteins. That's what happens here. Okay. The protein that gets made includes a replicase, which makes a minus strand, and that minus strand of mRNA acts as a template to make plus strands, so more copies of the virus. And then all this machinery that gets made by your own ribosomes, but coded for by the virus, that machinery makes more copies of the virus, and then the virus gets assembled into new copies of the virus called virions. Okay, this is a lot of vocabulary and stuff that I'm front loading, even though we're going to talk about what all these words mean later. I just want you to have a general sense of what's going on so that as we run into these things and I say to you, see, this is how this is involved in COVID-19, that you have some sense of what's going on. But if you don't get all of the stuff I just said, that's fine. We'll go over it many more times. There's an article that came out just this week that talks about the structure of the spike proteins on virus. And this is interesting from a cell physiology point of view and a biomechanics sort of point of view, but also from a practical point of view in terms of fighting this virus. If we know exactly what it looks like instead of approximately what it looks like, then you can use that as a strategy to design therapies to stop it from entering the cell. And I also want to mention that, so here's the reference for this article. This is what's called an accelerated preview. So this was accepted in the last week. It has been through peer review, but the journal hasn't actually put it in an issue yet, but they're making it available for people to read because it might help in terms of coming up with a therapy. Normally, you wouldn't get to see this for a few more months. There are other articles, and I'll mention one later in this podcast, where you get a preprint that hasn't gone through peer review. And that's more and more common in this pandemic, in terms of the academic literature in this pandemic, is that scientists are making their information available before it's gone through peer review. And that's in the interest of making science go faster. But you should just remember that because it hasn't been through peer review, they could be wrong and there could be big mistakes in there. But this one has been through peer review. And they came up with these images of the spike protein. You see lots more detail than in the standard spike picture that you see of the coronavirus. And one thing that's really interesting is that there are several joints in the stalk of the spike as it attaches to the virus. And that allows that spike protein to be really flexible and to be able to rotate. And why that's important is that when it's looking for a receptor, an ACE receptor on a cell, it might not have the exact spacing to where that ACE receptor is. And because it can bend and rotate, it can grab, so to speak, a receptor on a cell surface and get in to that cell. So this flexibility is thought to be important for how infective a virus is. And this is new information about this virus. The other thing that is just in the last couple of weeks is how modified this virus is with sugars. It's what's called a glycoprotein. So this is showing just the amino acids and not their carbohydrate modifications. This is with their carbohydrate modifications, and you can see it is a different shape with all those carbohydrate modifications, and there are a bunch of them. And why is that important? It's because if an antibody is going to attach to this protein, this spike protein, and by attaching, prevent the spike protein from binding to its receptor and therefore prevent it from getting into a cell, these glycoproteins, these carbohydrate additions to the protein, can prevent that antibody from binding or make it harder for it to bind. So the antibodies that I'm talking about are so-called neutralizing antibodies. And you can think of them sort of like a child-proof cap on an electrical outlet. So you don't want your toddler to stick their finger in an electrical outlet, you put a protective cap over it. If you don't want the virus to get into a cell, you can attach an antibody to it, and that prevents the virus from attaching to the ACE receptor. And these are the neutralizing antibodies that doctors are looking for, and that do people exposed to the virus make these antibodies? Can we make these antibodies after you've been vaccinated? Well, the antibodies need to see the real surface of the outside of that spike protein, and so knowing that these sugars are there may affect whether or not your immune system recognizes this protein. Okay. One aspect that we talk a lot about in another course, cell physiology, that has been very important for this pandemic is the technique called qPCR. This is quantitative polymerase chain reaction. This is the COVID test that most people are talking about when they talk about getting tested. This is they put a swab up way up far into your nose. Then what happens is you extract RNA from that swab. Remember, it's an RNA genome virus. You convert that RNA into DNA in the lab, and then you use this test to see, do you have viral DNA in your nose? And the way this test works is sort of looking for a needle in a haystack, in that you have some molecular probes, primers, short pieces of DNA that will bind to specific DNA and only that DNA. And you are trying to amplify that DNA. If you stick that probe that is sticky for a particular piece of DNA, in this case it's viral. So the virus has an RNA genome, but RNA is hard to work with. RNA tends to be unstable. So in order for it to survive the testing procedure, you convert it to DNA first, which is stable. So this test is testing for DNA, even though the virus is RNA. That's just a technicality. Okay, so you put the probe in, and when the probe finds its complementary DNA, which originally came from the viral RNA, it starts to amplify. And when it starts to amplify, it gives off fluorescence, and you get more and more fluorescence until you run out of reagents. So this is a typical sort of curve for a qPCR test. Okay, what does that mean in terms of how the test works? Well, that probe is looking for a needle in a haystack. And if you have a haystack that is a certain size and there's 10,000 needles in there, then the chance that the probe finds the needles is greater. In other words, it finds it sooner. So imagine you are looking for that needle in a haystack, and this axis down here, this x-axis, is how long does it take you to find it? Well, if there are a lot of needles in there, you find it soon on the left side of the curve. If there are not many needles in there, you find it later on the right-hand side of the curve. So when someone has a COVID-19 test, there's a cutoff, and that says that anything below this cycle number means that you're positive, and anything above a certain cycle number means that you're negative, because it's a very sensitive test. It can detect only one molecule in the reaction among billions of molecules. So it's possible that you can have amplification really, really late in the cycle, but there's not really anything there. Not enough for you to call it a positive test. Whereas if it's on this end of the curve and you find it early, then there's a lot of viral particles there, and you are positive. But it's somewhat up to interpretation as to what is negative and what is positive. So all these tests, they always run a control, in that they run a sample that is from someone that they know is positive, and they run a sample that they know is negative, in that, for example, it doesn't have any viral RNA converted into DNA in it at all. They don't put anything in there to know that their test is working okay. And then they test those against people tests. This takes a fair bit of time. Like we do qPCR in my lab all the time. Go through all those steps would take half a day. And this contributes to why testing is taking too long in the United States right now. Okay. There are other kinds of tests. So instead of this test, which tests is the virus in your body right now when I swab your nose, antibody tests says, have you been exposed anytime in your past? So do you have antibodies against the virus? You make antibodies against it from being exposed to the virus and your immune system reacting to it. So you're not detecting the virus, you're detecting antibodies against the virus. And this is the same as an HIV test. Do you have antibodies indicating you've been exposed to the past? But it's complicated in that if you do have antibodies against the virus, it doesn't necessarily mean that you're immune. So it depends on how many antibodies that you have. What is the concentration of antibodies? It depends on what kind of antibodies you have. Are they neutralizing antibodies like we just talked about? Now, we don't know the answer to these questions. And the studies just haven't been done. But there was a study that came out just yesterday about a fishing boat. And this fishing boat had a crew of over 100 people on it. I think close to 200 people on it. And they tested everybody before they went out to sea. And because they were going out to sea for several months. After only 18 days, they came back into port because they had lots of people sick on the boat. 100 of the 180 people, I think, was the number, were sick on the boat. Okay, so everybody was negative going out, but somebody obviously wasn't really negative. It's a false negative, and they infected other people on the boat, and they infected most of the people on the boat, over half. Very interestingly, though, the 2 sailors on that boat who did not get sick had a positive antibody reaction without any symptoms. They had evidence that they had antibodies against the coronavirus beforehand, and they didn't get sick. So it's a small study, but it is encouraging that in humans, if you do have antibodies, it's possible that they could protect you. So far, it's not ethical to infect people to find out on purpose, to find out if that's true. But here's a natural situation where they had all the data, and these 2 people had antibodies and they were protected. So the chance that they would have been protected anyway, or not developed the virus anyway, is pretty small because so many people were infected, and that they avoided infection might be related to them having antibodies. Okay, so that's one test, and they're problematic in that they're easy to do, but it's really hard to know what they mean because we don't know how long the antibodies are going to last, and besides that one story, we don't know if the antibodies are going to be totally protective. There are antigen tests where some part of the virus is detected using an antibody. So you develop an antibody in a lab that is against some part of the virus, and then you just test can I detect this part of the virus in a sample from this person? It could be a sputum sample, it could be a blood sample, whatever. This is the test that Governor DeWine took when the president came a couple of weeks ago, and it said that he was positive, even though the qPCR test said that he was negative. Another idea in terms of increasing the rate of testing is what's called pooling samples. So if you get 100 people that you want to test, you put all 100 people in one reaction, and you also save part of the samples that they give you for follow-up reactions. If all 100 of those people are negative and you pull them all together and it comes out negative, well, you only did one test versus 100 tests, and so you could save lots of time. This only works, however, if the population has a really low infection rate, in that it's unlikely that someone is going to be positive. It's more likely they're going to be negative. In states now where you have infection rates that are over 10%, over 10% of the population is infected, like in Florida or Arizona, pooled samples don't make sense because you're going to have to go back with every single pool and test everybody anyway. Okay, so false positive, false negative, what does that mean? So if you get a false positive test, that means the test is saying you've been exposed to the coronavirus, but that's not the truth. This is more rare than a false negative test. So if we're talking about the qPCR test, this is the one where when they're talking about testing in the United States, by far, this is the most common test. This is a really sensitive test, and it's very unlikely that you could be on this side of the curve if you really are not making the virus, or if you're not infected with the virus. Sorry, it's very hard to be positive if you're not infecting the virus. You could maybe be out here on the edge of being negative, and it's sort of a judgment call. That's a possibility. But if you get a strong signal from a qPCR test, it's pretty reliable that that sample has coronavirus in it. So false positives are more rare than false negatives when using qPCR. But when using the antigen test, that's based on an antibody, they can still show up. And that's what looks like to have happened with Governor DeWine, in that he had an antigen test. That's what the president's team was using because it's really fast. It said he was positive, and then subsequently he had 2 qPCR-based tests that were negative. And he's never shown any symptoms, and so it's very likely that he truly is negative. What would cause a false positive in an antigen test? So that test I looked up is using a protein that is not the spike protein, but another protein on the outside of the coronavirus. It's possible that that other protein that is on the outside of the coronavirus is similar enough to some other coronavirus that the governor was exposed to maybe as a kid. So he had a cold when he was a kid. It was a coronavirus caused cold, just not this coronavirus. And that other protein caused him to have a false positive with this test. Okay. False negatives are lots more common, where it tells you you have not been exposed to the virus, but in truth you have been exposed to the coronavirus. So why are these more common? In that it's actually a pretty short window where people are infected that they will test positive. And there's countless stories about this in this pandemic, where people will be in the hospital with symptoms, fever, shortness of breath, bacterial infections in their lungs, secondary to a viral infection, and they're still test negative. And then they keep on testing them because it doesn't make sense that they're negative, and then eventually they'll test positive. So there's actually, during the incubation period when someone is infected with a virus, there are days where the virus is present in someone, but there's not enough virus present to show up on this test. Or it's in a different part of the body than is accessed by this horrible, nasty swab up the nose test. So negative tests are false negatives are common. If someone has a truly negative test, that it only gives you what their history is up until that point. So if I have a test today and it's negative, and that's a true negative, not a false negative, and I wouldn't know, it tells me that I have not been exposed to the virus up until the day that I'm recording this podcast. But it doesn't tell me 10 minutes after this podcast is over that I couldn't be exposed to the coronavirus. So it's not a excuse to go party it up with you and 10,000 of your favorite people. It just means that you've been lucky up till now, and stay vigilant. Okay. In the United States, the U.S. is testing about 750,000 people a day, and Ohio is testing about 20,000 people a day. And you can go to Johns Hopkins University using this link to find out how many people are being tested per day and what is the positive rate per day. That's not enough. That sounds like a huge amount, and it is a big effort, but it's not enough testing to adequately control the pandemic. So, in order to be able to control the pandemic, you've got to be able to get a test without waiting, and you've got to be able to get your results in enough time to have some meaningful action as a result of getting those results. So as I'm talking about this now, most people can get a test within, say, a week in Ohio, and then about another week to get the results. So that's 2 weeks after they're worried that they might have been exposed. And that's way too long, way too many days to be adequate. So if someone is being super responsible and they think they've been exposed, they go have a test and then they sit for 2 weeks waiting for the result of that test and aren't in contact with anyone. That's a big burden to put on someone and saying, don't work, don't go outside, don't be exposed, don't expose anybody else for 2 weeks. That's difficult. And that's what some countries are asking of people when they travel to the country, is that they quarantine for 2 weeks. If they're positive in that 2 weeks, they're going to need help sooner than that 2 weeks in terms of trying to get therapy. Just 2 weeks is turnaround is just way too long a time for us to get control of what's going on.